Welcome to the After On podcast. I'm Rob Reed.
And I'm Tom Merritt. And this is a special episode designed around the book After On.
Each of these special episodes focuses on one aspect of the science, tech, and social issues explored in the novel. Now, you don't have to read After On in order to learn from or enjoy these podcasts.
That's because we wait until the very end to discuss the book in detail and to really tie it to this week's topic.
That last section will make perfect sense if you're reading the novel. But if you're not reading it, just tune out at that point. You can always come back if you decide to read it later.
And we'll warn you before we get to that part. But first, we have a lot of things to talk about, which should be interesting to everybody. Specifically, DNA printing. And by DNA printing, we literally mean the idea of a printer that can print out DNA that can be used in living organisms. If you're thinking, Tom's talking about the novel now, well, no. At least not entirely. This is one of those odd situations, Rob, where the truth is just as strange, if not stranger, than the fiction you wrote.
Quite possibly. I sketched out a couple of completely plausible scenarios, fictitiously and After On, but our guest, Andy Hessel, describes even more possibilities, many much more positive than the ones that I used in the book. So let's talk with Andy Hessel about synthetic biology and what could actually go right with it. Andy, thank you so kindly for having me up to this beautiful spread of yours in the town of Cazadero, California. Did I say it properly?
You said it correctly.
And
welcome.
It's towering wet redwoods and beautiful weather. And we are going to talk some synthetic biology. I'd like to start with your background. If I'm not mistaken, you've been on leave from your PhD for a couple of decades now. Is that correct?
That's about right.
And so let's talk about where you were in school when you launched into this post-school hiatus that you were still in.
I was just a geek. I loved just about everything mechanical, electrical, or electronic. I had started using computers quite early in the early days of the personal computer. But then when I sat down and really started to get serious about where my life was going, I realized I'm not really interested in programming computers. I'm more interested in programming biology, and I switched to life science. And it was a really good time because genetics just started booting up. In earnest, the Human Genome Project got launched, and we started to get a lot of digital information starting to come into the world of biology. And none of the scientists had ever worked with computers.
You were in Canada, and then you had come over to go to the University of Illinois at Urbana, correct?
At Urbana. So the first 5 years of the Genome Project, from 1990 to 1995, I was still in Canada and largely working in the field of bioinformatics.
Yeah.
I was reading and analyzing bacterial genomes. Mm-hmm. And I really pushed the limit of what I could learn in Canada at the time. And so I was going to move to the United States, University of Illinois at Urbana-Champaign and continue my work there.
And that was a really interesting time there because that was right after the Mosaic browser, the great-grandfather of every web browser we know, was invented by a bunch of undergrads at University of Illinois at Champaign-Urbana, right? It was Mark Andreessen and his merry band. And there was also a huge supercomputing center there, correct?
Yes.
Was there also a major life sciences set up there?
Yeah, they had one of the largest grants at the time for sequencing. So they were producing millions of base pairs of genetic code.
Oh, really? Were they part of the Genome Project then?
I'm not sure what the direct involvement was with the Genome Project, but I know they had one of the largest grants, and they had just vast amounts of genetic data at the time coming in that needed analysis. They also had a fellow that I was really fascinated by, a guy named Carl Woese, who had discovered a whole branch to the tree of life called the archaea.
That's number 3, right? Branch 3
There's
is 3,
prokarya,
right?
the eukarya, which is you and me and all the other mammals, and then there was the archaea. So just a fascinating branch really early on of these ancient microorganisms.
And he was there. So you went there, you registered for your PhD program. Did you even complete a day of study?
I got scooped up by the pharmaceutical company Amgen.
And so you went to Amgen ultimately instead of the University of Illinois.
That is correct. They made me an offer I couldn't refuse.
And then fast forwarding way forward to where we are right now, you're now at Autodesk. Autodesk is known for making the most, I believe, the most sophisticated design tools, computer-aided design tools in almost every field, really. Whether it's computer-aided design writ large, computer-aided manufacturing, architectural tools, a lot of the Hollywood post-production tools come out of Autodesk as well. Is that correct?
That is correct.
And so merely because you're there, logic would dictate that Autodesk is making a very big bet on designing tools that will help people design DNA sequences, design molecules, design life from the ground up. Is that essentially the effort that you guys that you're personally engaged in there?
Yeah, that's the general idea. Visualization, simulation, and manipulation tools for this nanoscale world and the world of living systems, which are, when you think about it, they're self-assembling. Like the beautiful part about life is it takes elemental materials and a little bit of energy from sunlight or sugar, and it builds really complex things like trees or you and me. And so, but that process, it's a manufacturing process, is very complicated, and it's best managed in software.
In the midst of all this, I think it was early 2016, you instigated, for lack of a better word, the start of Human Genome Project 2. Is that correct?
It was called HGP2 when we first started doing it. It morphed into HGP Write, distinguishing it from HGP Read. And because of pushback and just listening to the community, we dropped the H out of it because it's not just about the human genome.
Okay.
It's about being able to understand and write all genomes. Mm-hmm. So it's now called GP Write.
And there is a notion, though, and correct me if I'm wrong, of building a human genome length strand of DNA ultimately, correct?
Exactly. It's about being able to make large genomes ultimately, like the human genome, which is 3 billion bits of code. And that is a several orders of magnitude more than what we can do today with current technology. So it's about setting a high bar and creating this pull of scientific interest and ambition and inspiration to be able to do essentially much more than we can do today.
And GP Write has other goals too, right?
There's multiple goals of the project. A lot of it is just to engage the community and the public in about these technologies of being able to design and build, literally code living organisms.
Yep.
So there's certainly a big awareness aspect and engagement. It also provides a venue to discuss complicated things like intellectual property and ethics, and really just scientific directions. Mm-hmm. And then there's an engineering component. We need better tools and software and DNA synthesizers. And then there's pilot projects, which are scientifically interesting. And this is starting to build organisms that have useful features, which can be either benefits for academia and research or for industry.
So GP Write is all about writing or synthesizing DNA. But before we can write, we need to learn how to read. And that's what the Human Genome Project was all about, reading the DNA of an entire human genome for the first time in history. And it took 13 years. Now, of course, you know this, Andy, but for the benefit of those who don't, in DNA, the fancy word for reading is sequencing. There's synonymous terms. Sequencing just sounds much smarter. So if you read a genome, you say you sequenced it because you determined the sequence of the DNA letters that comprise it. Now, I'm making a point of all this because we're just entering the era of DNA synthesis, and the things that happened to cost and performance during the sequencing revolution should be predictive of what's about to happen. So let's look at what happened to DNA sequencing during its emergence. The Human Genome Project was 13 years, $3 billion roughly for 3 billion base pairs in the human genome, netting out to an incredibly remarkably, almost suspiciously round number of about $1 a base pair.
Yes.
So now today, Veritas Genetics has been offering a $999 sequencing package now for one's entire genome since March of 2016. So to go from $1 a base pair to, I think that pencils out to 3/100,000ths of a cent for a base pair, it's way faster than Moore's Law. And we all know how transformative Moore's Law has been. We've all lived that. Why is it that it has come down so rapidly, so much faster than even the transistor? What has driven that?
The capabilities largely grew out of some incredible advances in microfluidics, in electronic technologies, and robotics. That's what's really powered it. There's also been some more biological advancements. Today, most people don't realize this, but we actually sequence DNA by leveraging the process of writing DNA that's used in our cells.
Yeah, it's called sequencing by synthesis, correct?
Yeah, and it's often put under this banner of next generation sequencing, which is really silly. It sounds like an episode of TV.
Well, it also sounds very final.
Yeah.
Oh, it's now the next generation stuff. How do you talk about that 5 generations later?
Right. We're actually on the 7th or 8th technical generation of DNA sequencing technology. So it's been evolving, and it's been evolving incredibly quickly because it is so foundational.
Now, was it foreseen? Because one of the interesting things about Moore's law is, I think Gordon Moore first articulated it in 1965 based on a very small set of data points. But after it had lasted some period of time, people were very, very confident predicting that this is going to last indefinitely. Early on, let's go back to 1990 at the start of the Human Genome Project. Was there a realization that we're going to push it down that fast and that violently? Or were the participants in this project astounded by how quickly sequencing got cheap?
To give you a frame of reference, we didn't even sequence the first bacterium, approximately 5 million base pairs, until 1995, which is halfway
into it, almost halfway.
Essentially, a third of the way through the Genome Project. But when they had done the forecasting for the Human Genome Project, they really needed a ballpark number. So they estimated about $1 a base, knowing the genome size was about 3 billion bases. But they also factored into that the cost would fall over time, because they knew that they'd get better at doing this, that automation would come in place. And so we saw this technological evolution that continues to this day.
There really is no looming brick wall on the horizon. We can expect the cost of sequencing to continue to plummet for the foreseeable future. We're not getting to any outer limits of physics or capability.
Absolutely. The technology will continue to be refined and improved. It will change for humans, though, because we're already, reaching a point where the human genome is, as you pointed out, sub $1,000. I'm already seeing evidence that in the next while, it will drop to free. Because new business models are appearing where the cost of sequencing can actually be pushed off to essentially other individuals and organizations. Because remember 20 years ago when Hotmail came out?
Oh, yeah.
Hotmail was radical at the time because they gave away free email accounts, whereas everyone up to then had to pay for it.
Yes.
Today we take it for granted. We're at that point where soon people will give away free genomes. And beyond that, I expect that some people will actually become millionaires because of their genomes. People will walk up to them and say, you have a feature that I just think is incredibly attractive, and I'll give you $1 million if I can get a license to it.
Really?
Yeah, that's coming soon.
That's interesting. Well, actually, there are two very interesting things here. I want to first talk about the free genome. Then I want to come back to the $1 million genome, which is also pretty fascinating, particularly if I happen to have one. Illumina mentioned or has announced with NovaSeq, which is their new equipment, that they're going to deliver a $100 genome by the end of this year.
Yes.
If they're on time. We've come down another order of magnitude. What is interesting about that is you're starting to get into the acquisition costs of businesses, right? Businesses pay $100 to get a customer all the time, but that's not an unreasonable acquisition cost in healthcare. And so, yes, now a company can give away a genome to get a customer. That's a pretty good deal.
Especially because there's a lot of information in a genome, and it can relate to a lot of different parties. So one customer doesn't have to pay for it.
Right.
You can actually divide the cost up amongst 100 different parties and pay $1 per customer.
I think the Hotmail analogy is actually a very astute one, because that was a tipping point enablement as well. It was just that Moore's law had moved so fast and bandwidth was coming down so rapidly, and disk space was the real big thing when it came to providing email, and all those things had just barely gotten to the inflection point where they could do that rationally. And they may even have been a little bit ahead of the curve in terms of economically rational. They were able to access capital at late 1990s prices, which was very cheap capital because we were in the internet bubble. Has anybody started doing this yet? Has anybody started giving away free genomes or free sequencing packages?
I haven't seen free yet, but there's so much fuel on the ground. There'll be a spark, and you'll suddenly see an organization with millions of genomes in their databanks.
So let's talk about this other thing that you mentioned that's intriguing. If somebody has a quirk to their genome, first of all, how might that be very valuable and to whom? And secondly, I assume that because you mentioned that, a person who has a quirky, unusual genome in a sense, owns that as intellectual property. That genome cannot be exploited or the data that it generates manipulated without that person's consent?
Well, I think some of that still has to be worked out. But if it's your genome and you have a particular feature and someone wants to analyze that feature, they're going to approach you and ask you for a sample. If they perceive it as valuable and they're willing to pay for it, I'm absolutely certain these types of contracts and arrangements will be made. And so many people have superpowers. Yeah. Like so many people. I have met people that have super hearing. I have met people that can almost see in pitch darkness. Some people never lose their hair. Some people never, their hair never goes gray. Some people don't have facial hair, and so on and so on. They're very valuable traits.
So do you imagine somebody would identify somebody with an exceptional trait, make the leap that it's perhaps genetic, and then say, hey, I'm willing to give you X dollars in order to find out if it's genetic, or I'm willing to give you X dollars if in fact it proves to be genetic?
Yeah, give me a sample of your genome. I'll give you X number of dollars, and perhaps even a percentage of any products brought to market based on that genome, for example. And who knows what these contracts will be, but I absolutely expect that someone will become a millionaire simply because of the code that makes them.
Not if they gave their genome away to somebody for $100, which is another interesting reason why somebody might want to give away.
It depends on the end user.
It depends on the EULA. Everything depends on the EULA. But I would imagine that I think the guy's name was Sabir Bhatia, who started Hotmail. I imagine the Sabir Bhatia of genomes is going to be really smart about the EULA. I will be astounded if it's not essentially free to get sequenced in a lot less than a decade, and perhaps not much more than a year.
I expect the tipping point now to be within the next 18 months.
Yeah, and I would not be at all surprised to see that. So now let's talk about the synthesis curve. What does it cost to synthesize a base pair today? And I know that's a complicated question because it's dependent on length. So let's start with a relatively short oligo. Like what Twist does,
let's just explain what an oligo is. Yeah. DNA isn't synthesized in one big giant string. It's actually synthesized in short fragments, typically under 100 base pairs of genetic code, and then it's assembled. So you can actually synthesize in oligos an entire human genome, billions of base pairs, for a few $1,000s. But the assembly of those oligos into longer strands is incredibly expensive.
So you'd have something on the order of, I guess, 30 million 100 base oligos that you would then have to strand together.
It's the stringing together in the assembly process. Yes. Because DNA is not typically made using a chemical process and an assembly process like this. That's a human invention.
Yes. It's not typically made in our bodies using the oligo assembly process.
Exactly. Yeah. That's a human invention. Right now, I would expect if we were to synthesize a human genome using this approach and assemble it, that it would cost, assuming some economies of scale, it wouldn't be $1 a base pair today. My best estimate would be about $0.10 a base pair. I would expect it to fall between 100 and 300 million if we brute forced the synthesis of a human genome.
And the brute force, again, just to review, would be making short strands of 100-ish, making 30 million-ish of them, and then joining them arduously one after another. That would be hundreds of millions of dollars, but not $3 billion.
So not $3 billion. We're already past that $1 a base pair point. But if you're making shorter segments, a single gene, which encodes essentially for a single protein, is typically between 1 and 2,000 base pairs. You'd look between $100 and $200 to make a gene. I remember in around 2000, if I was to write a single base pair of genetic code, it would be about $13 to $20.
$13 to $20.
Right. And now we're paying
a dime
or less.
Going back to, do you brute force that now? Let's say we have a gene that put it right in the middle, 1,500 base pairs. Is that made by stitching together 1,500 base pair oligos? Is 100-ish?
Yeah, essentially.
Okay. Yeah. So 100-ish is roughly the unit of production right now. Is that fair to say? Yeah. And how error corrected is that?
Well, that's a different question. The error correcting processes are different with different companies that do this work. Some of them leverage biological processes, as well as you have to sequence and verify the result after the assemblies. So, but they've gotten very good at these processes for gene size fragments. When you start building larger fragments, say 10,000 base pairs, now you're having to string together multiple larger gene size
fragments. Yeah.
Right. So the process there it is longer, requires more validation, and is going to have more errors as you start to stitch this together. But if you look at building a circuit, if you look at the gene as a component, when you start stringing them together, you're essentially building a metabolic circuit.
Yeah.
A lot of companies today that make these metabolic circuits and they start doing the reprogramming of metabolism typically work in 50,000 base pair ranges, and their systems for doing assembly and error correcting and validation are really fairly well refined.
Yes. For
the 50,000 base pair size. But that is essentially requires a large investment of robotics and technologists to be able to do that type of work at scale.
These are folks that are building basically metabolic circuits out of a handful of genes. And generally, they're producing some kind of a substance, right? Like industrial feedstock. What are the types of things that get produced at that point?
At that point, it's often materials, things like industrial enzymes, which may be used in your laundry detergent, for example. It can be a medicine that requires multiple enzymatic steps to make the medicine, because organic chemistry done by enzymes is magic.
Yeah.
So various materials, polymers, could be a biofuel. It could be an antibody. So there's multiple things that can be made at that scale, which is why we're starting to see companies like Ginkgo Bioworks in Cambridge appear that are really focused and have a foundry for doing metabolic engineering.
And so these are some of the customers of a synthesis company. Somebody who is putting together a metabolic circuit, let's call it 50,000 base pairs. Let's call it a couple dozen genes to create something. They have very, very carefully studied how these genes could work in concert in order to output something of value. Now they're going to go to somebody who creates DNA and say, I need this 50,000 base pair string, and that is going to be made for them. Now, a major maker is Twist, correct? Twist Bioscience?
Yes.
And they are basically synthesis as a service right now, right?
Synthesis as a service. Their processes today are optimized for gene size fragments, but as their systems get more sophisticated, they start making and being able to deliver larger size fragments.
And so, not to try to explore the entire domain of the synthetic biology industry, which is quite large, but they are a major player. Twist is somebody that supplies lots and lots of people with their synthesis needs. Now, if we look at a comparable company in sequencing and DNA reading, the giant there right now is certainly Illumina. They're not sequencing as a service, they're selling boxes.
Yeah, boxes for preparing DNA, for sequencing the DNA. They also have tools for analyzing the DNA, storing the data that's generated.
Yeah. So given that Illumina is long past the stage of being sequencing as a service, do you imagine that synthesis will follow a similar path? Right now, sequencing is a very distributed process. People buy the boxes and they do it all over the world. Do you think synthesis will follow a path as well? And the Illumina of synthesis will be a manufacturer of hardware that other people use?
So when it comes to synthesis, I think it'll actually go distributed fairly quickly. Right now, it takes a fairly large, complicated box to synthesize DNA. It takes robotic tools to assemble the DNA. You have to make a fairly substantial investment just to get to that 50,000 base pair range.
And that is Twist and their competitors are those who have done that.
Well, and Twist has raised several $100 million to do this process at scale, reliably, at low cost. That being said, DNA synthesis and assembly is something that happens in the world around us.
Yes, you and I are both doing it right now.
Right now.
Yes.
In little factories we can call cells.
Yep.
And they do it using a different set of chemistries that are exceptionally fast and reliable and cheap and capable of synthesizing and assembling entire genomes. So I actually think that if I were to leap ahead in the future 25 years, I would be really surprised if I don't have a DNA synthesizer in my lab or in my work That's or in my pocket.
Yes.
Like it will be, it'll be a desktop device or smaller, and also very inexpensive synthesis services if I choose not to have any devices.
Yeah. Now, as we go up the chain, bioprinting is still a little difficult. We're not able to synthesize DNA at the length necessary to enable that just yet. What are the breakthroughs that need to happen on that front in order for that to come forward in a big way?
Oh, those are multiple areas there. We need higher resolution and faster printers. We're getting that just from the traditional 3D printing world.
Yep.
You also need better cellular inks, because living cells don't necessarily like to live in free solution. They're alive. You can't necessarily have them sitting in the printer for a long period of time like your inkjet. So there's a variety of different problems, and learning how to put cells together so that they function as a tissue or an organ or even an organism, it just takes a lot of monkeying around with bioprinting as a foundational technology. But it's one of my favorites.
Yeah, interesting. What do you think one of the first really revolutionary applications of bioprinting is likely to be? Something really useful, whether it's on a health level or an industrial level?
Well, we're starting to see it on an industrial level already with the manufacturing of sheets of tissues that can be, for example, like leather. Leather is a sheet of tissue, but you don't necessarily have to make it from an animal today. You can print it in large sheets and any shape and pre-pattern sizes as you like. Foods in many ways are just cells, or biological products that are not only, made of these different compounds, but tasty. And so now you can actually make your food in different shapes. So we've seen bioprinters be used to make things like bone. Because the cells that produce bone are relatively easy to print, and then they essentially make the bone around them. That's pretty interesting. But I think the one that I'm waiting for, the one that I think will be the one that kids can play with, is just the group that figures out how to make a muscle solution, essentially cells that can be printed and innervated with a little bit of electronics. And now you can start making little robots, because that's all muscle does. It just contracts. And so now you have essentially a little motor. So I love the idea of just having 3D printed muscle cells.
That is interesting. So sort of a muscular Lego.
Yeah, a muscular Lego. Now you can build little robots. Think of an insect, and just, they're powered by muscles, but it doesn't take a lot of cells to generate, the force to move a spider leg, So I just love that idea of being able to make little robotic insect-like creatures with my 3D printer, and just having a little electronic chip powering all the muscle motors.
Is that a coming decade thing, or is that deep future science fiction?
I don't think it's deep future science fiction. I think it's really just the right people working on the right problem and finding a little application for it. But it doesn't stop from there.
Yeah.
So that's just reprogramming a cell. But what happens when you now have the synthetic capacity to make an entire genome? So a bacterial cell genome, like E. coli, is about 4.5 million base pairs.
Yes. Let's call that maybe almost 100 times the scale that we're operating on right now.
Right.
If we've got an enzymatic circuit, if we've got a metabolic circuit, rather, maybe that's 50,000 base pairs. So getting up to that level, that's a leap of roughly 2 orders of magnitude.
Right. So if all of a sudden being able to write 4 or 5 million base pairs is $100.
Yes. What do we start doing then that we can't do now? What do you think the applications will be when we get to that scale?
Well, now, so let's just say our imaginations will have to grow.
Yes.
Because now we can design entire free living organisms at the bit level. Mm-hmm. So we can start to program in much more complex features into the metabolism of the organism. So we can have it turn on or off with different stimuli. We can control the carbon flow of the metabolism, essentially the energy flow through the organism, with much higher fidelity. We can do much more research, because now it costs less to fail, because we don't know all the rules of programming biology. Evolution didn't keep notes.
Yes.
So we have to reverse engineer a lot of these paths. But as it gets cheaper, now we get to write more code, we get to fail faster and cheaper, and we get ultimately to build libraries of components, parts, and systems that we can start to assemble to make more complex features.
Now, going from where we are right now, from metabolic engineering to full bacterium, 2 orders of magnitude in terms of the length of the DNA code, How many years roughly does that feel like, given the price compression curve that we're now riding on the synthesis side?
So we can actually predict this fairly well because we've got a number of years of data on the price compression of DNA synthesis. And it's absolutely exponential. The costs have been coming down. Again, I've said it went from about $20 a base pair in 2000 to a few pennies now.
Yes, which is extraordinary.
Yeah. But it still has a long way to go, as sequencing has taught us.
Yes.
But I expect to see a giant increase in performance, in price compression in the next 5 years.
And what will drive that? Will be sort of a paradigmatic transition, because currently Twist is hyper-optimizing chemical synthesis, correct?
Exactly.
And will this be a move away from that the chemical paradigm to something entirely different that's going to drive this move forward?
I think we're going to move away from the chemical paradigm of synthesizing DNA to the enzymatic synthesis of DNA, essentially leveraging the processes that exist in our cells today.
So how would that work? Tell us briefly how enzymatic synthesis would work and how that compares to chemical.
Sure. The cycle time of a chemical synthesis process is, let's be generous, let's call it a minute. Mm-hmm. That means every minute you can add one new element to the DNA chain, one new chemical bit. Mm-hmm. And you can do this in parallel. So that's the secret of Twist's capabilities. They do this in
Twist, the company.
They do this synthesis in a highly parallelized way. But again, you've got all these downstream assembly problems. When in our cells, a single enzyme molecule called a polymerase writes DNA, and it writes it by using the as you know, DNA is a double helix. They unwind the helix and it writes the DNA using the unwound DNA as a template. Mm-hmm. But forget about the low-level mechanics of that. The enzyme itself is actually biochemically assembling the DNA chain, and it does this at about 1,000 chemical bits per second.
Wow.
And it does it with an error rate that's negligible. Otherwise, we'd all be dead of cancer.
So literally at about 60,000x the rate of what is currently happening in a state-of-the-art facility.
Yeah, it's just incredibly fast and accurate. So, and it can also be parallel, done in parallel. So if we can reach into that cellular machinery and pull out these polymerases and direct them how to write DNA, what DNA to write, we essentially have these little processors, little factories for DNA that are now controllable by using computer systems.
So currently they are copying a template, and the challenge would be, in order to harness them in their speed, would be to basically say, you're no longer copying a template, you're now just spewing out the letters that we have designated. And how we direct the polymerase activity is where I think some of the most exciting research is being done today, because there's a variety of ways you could do it, and one of them might be electrical control, just finding some way to build an interface to the enzyme and say, this is what you're going to write, just using chemicals, electronic signals. Another one might be optical signals, having the polymerase write a particular base when you shine a certain wavelength of light on it. So I zap it with green light and it writes a G. I zap it with red light, it writes a T. And if you could put something together like that and move at the pace of the enzyme, then all of a sudden, instead of it copying a template, you're dictating to it, and it's taking down the dictation.
Telling it exactly what to write. And again, light, you can responds very, very quickly. Yes. So these are two possible paths to making a programmable polymerase.
And that would be shattering the speed records that we have right now.
Yeah. So just do the math. This was all done at a molecular scale. Mm-hmm. So you could have an array of, say, 1 million of these polymerases on a chip. And sending the signals to these polymerases is trivial. Yeah. But if you have 1 million polymerases on a chip, you'll be writing 1 billion base pairs of DNA per second. Yeah. And this DNA will be virtually error-free. Mm. So we don't know how long a strand we can actually make. So I can imagine chips that are writing DNA and then being sequenced in real time. The DNA being written is going through a nanopore sequencer, essentially the reverse process of using a biological structure to interface to a computer to read the DNA.
Yeah.
And then having some sort of a way to stabilize that DNA so you can write, say, 1 million base pair strings without it breaking and fragmenting.
So this could conceivably travel down the price performance curve even faster than sequencing did.
Exactly. Like we might see, again, 10 cents a base pair today.
Yeah.
And in a few years, essentially free.
Essentially free.
Like being able to write, again, if you're using 50,000 base pairs as kind of the basis for metabolic engineering, you might be able to make 1 million of those in under a minute on using one of these chips.
And what then will be the applications that will come into reality at that point? Is it just such a broad and crazy diversity, it's hard to even speculate?
Yeah.
Or are there things on the near-term horizon that you can say, boy, if we could only write 1 million base pairs as quickly as we can currently write 100, we would be doing this in an instant.
For me, it's a lot like digital computing. When it becomes that accessible and inexpensive, you just see this incredible acceleration of creativity because more people can get into the space and start to play with it.
Now, as we've been going through this revolution in ground-up creation of DNA and synthesis, there has meanwhile been a revolution in editing with CRISPR that has certainly got its share of press over the last couple of years. Could you describe CRISPR briefly and why it matters so very much?
Yeah, CRISPR has just turned out to be a really powerful new tool that's appeared in the genomic space. As we've discussed, we don't have the technology today to synthesize whole genomes like the human genome.
Yeah.
But we do, CRISPR allows us to make edits to that genome really easily.
So you can work with very large DNA strings that are pre-existing if you have CRISPR because you can edit them really robustly, even though you couldn't write something that big.
Exactly. Now we can do essentially search and replace on that large genome and add in a new feature or turn off a particular gene, etc. It's a really powerful and inexpensive tool. And so this is, in the last 5 years, we've seen this tool appear. A number of companies and research programs appear just to explore the applications of the tool. We've seen applications in gene therapy, in cancer therapy, in vaccines, and more. It's remarkable how quickly it's caught the attention of investors and of researchers.
How much more quickly and reliably does CRISPR enable a very, very surgical edit to be made versus what it would have taken pre-CRISPR, which really was just 3 or 4 years ago?
Yeah, and we've always had editing tools, but they've just been cumbersome and slow.
Zinc fingers, I was one,
and they've worked, but they just haven't been as useful as CRISPR. CRISPR turns out to be you can use it in almost any organism. Mm-hmm. It's really expensive and really accurate. Now they're still verifying, you're still manipulating the DNA molecule in a living cell. And that always requires a lot of, searching for off-target effects and just making sure the biology is working properly, because biology isn't like computing. Yeah. It's always squishy. Things don't always work with perfect fidelity as we expect with computing systems. Yeah. But you can get really, really close. So CRISPR is really good at that. But at the same time, I think it's a transitional technology. Mm-hmm. For the work that I'm doing already in small genomes like viruses, it's too crude. Mm. Why should I use a tool that edits the physical DNA molecule when I can edit the DNA molecule using computer software and just print it?
You're working with strings that are small enough that you can write it from the ground up.
Right.
Whereas people who are using CRISPR are going to be working with mammalian DNA with much longer strings that cannot, but when they can be written from the ground up, there will be no further need for the editing.
There will be less and less need for editing.
So it's a transitional.
So
it's a transitional technology.
And there will always be applications where it's useful. Yeah. But the better we get at writing large genomes, the more we will do that editing and design work using computer software rather than manipulating the living organism's code itself.
Let's go back to, is it now called GP Write?
It's called GP Write.
Got it. So it was Human Genome Project 2, then it was GP2, now GP Write. But it's essentially the same.
It's essentially, just think of it as we're going to write human genomes.
We're gonna be writing stuff. Now you recruited
and everything smaller.
And everything smaller. So you recruited George Church early in the process to this, correct?
Yeah. George has been involved in almost every major genomics breakthrough since the Genome Project. He was involved in the first Genome Project, in the early 1980s. He is just one of the most well-known and funny and smart individuals in genomics, and has helped launch dozens of companies, and is just one of the kindest scientists I've ever met. When I called him up, I said, George, this is really starting to frustrate me that scientists haven't come together. Would you consider leading this project?
And George is also has been very core to the whole CRISPR effort as well, correct?
Yeah, absolutely. He's one of the co-inventors.
Now you have, it's hundreds of scientists from dozens of countries now that are part of it. Is that correct? Or about 100?
Yeah. At the last meeting, we had about 200 people from over 10 different countries, I believe, show up. The level of interest in this is remarkable. And people just realize it's inevitable. And now's the time to really start thinking about all the bits and pieces, organizing the scientists, organizing the private relationships, start thinking about the intellectual property, think about the ethics and the societal implications.
And I think you had said earlier in a prior conversation that the goal is to be able to synthesize a human scale genome by roughly 2026. Did I remember correctly?
To actually synthesize a human genome or other human scale genomes, and to install it in cell culture. So if I write a human genome today, people immediately think, oh, you're going to go make designer babies.
Right.
No, you can actually write a human genome, put that genome into a cell, and just grow that cell in culture and make just say human fibroblasts or human stem cells that can go on and become bioinks for your printer or produce a drug or etc. So right now the group is being scientifically led has been collecting project proposals for really interesting engineered human cells and smaller genomes. You don't necessarily have to do a human cell. You could prototype other smaller organisms. And so we've just been collecting a tremendous number of research proposals to make really interesting cells.
I assume that 2026, that's based on an extrapolation of how rapidly synthesis technology is getting better, how rapidly it's getting cheaper. If you did sort of a straight line analysis and you just assume that synthesis costs continue to come down at roughly the rate that they have been, what would it cost to make a human scale genome in 2026 ballpark?
Yeah, ballpark, it's under $10 million.
Under $10 million.
Yeah.
And in that year.
Yeah, in 2026. So this is why I believed it was so important to start the project now. Even if you don't agree with it, even if you don't understand it, even if you think that this is, it's too early. Well, you can make that argument about the first Genome Project.
Sure.
But just doing a straight line analysis, we can expect that, again, when something becomes under 100 million, it becomes essentially affordable by companies,
by many
individuals these days. So, but when it comes to 10 million, again, by 2026, this is really inexpensive, and then just about anyone with the will can go and do it. And so I felt it was really important for the scientists to take leadership again.
And the advantage of a consortium of scientists doing this first is that a great deal of the information ends up in the public domain. I assume that's part of it.
Well, and it's not even, this isn't like reading of DNA, which generates a lot of information that then you kind of have to sort out. This is about more building the design tools,
the processes,
and the infrastructure and the processes to go and assemble bigger and bigger genomes. And if we can do that in a pre-competitive way, then we can kind of stop focusing on who's going to have the next best device or the new best tool. We can start thinking about, well, what can we make with biology that's going to deliver the most value? And there's essentially an unlimited number of applications there.
Now, one of the things that intrigues me when I look at technology moving forward at this extraordinary speed is, who is capable of doing what? To put this into context for us, because you told me an interesting story about Craig Venter. when he was competing with the Human Genome Project for all intents and purposes in the late 90s. He took you on a grand tour of, I think it was one floor of his facility, and said, this is the world's second largest sequencing facility because the largest one is upstairs or downstairs. What was the scale of that operation?
It was really impressive. There were hundreds of robotic DNA sequencers installed in a single room. And it was a factory. It was
Factory
the first,
size.
it was the first factory I had ever seen for doing DNA sequencing at that scale.
And that capacity is now literally in a desktop system.
But now you can buy the factories, the machines that do this are
Yeah.
smaller and much higher throughput. So that Celera factory could probably fit in a very small space today.
Now, by contrast, let's imagine the tech who's operating the Illumina NovaSeq system at the end of this year, which can sequence a human genome for about $100 in cost. What is the training probably of that person? It's a lab tech, I assume, who has got a NovaSeq box, is very, very fluent in using it. Is that somebody with a bachelor's degree in science? Is that a really, really smart person with a high school degree? Is that a PhD? Who is the person who's operating that?
Oh, it's closer to the smart high schooler and bachelors. Yeah, like this is a process that's just, it's loading the machines and babysitting them.
So because of the forward march of technology and of all the research and wisdom that got embedded in that box, we are literally at a situation where a really bright high school graduate or a college student can essentially do the work that it took the entire life sciences community to do in a 13-year period not that long ago.
Correct.
So we're getting to the point where even teens can do things that are extraordinary. That's in sequencing. Presumably, synthesis is going to follow a similar path. So just as a lab tech can now do what the entire life sciences community had to collaborate to do a couple decades ago, we can assume at some point in the nearish future, a single really smart person with sophisticated equipment that does not yet exist will be able to do things that George Church and the entire GP Wright project couldn't do today.
Yes, I think that's a fair assessment. Now, I think it'll follow a trend not so dissimilar from 3D printing.
Yep.
Where you can have very powerful design tools and just when you're done, hit print. Because a lot of the hard work about understanding the nuances of the genetic code and designing cellular metabolism will just be encapsulated in the software.
Yeah. And let's talk about what brilliant teams are doing right now, because it is kind of mind-boggling. I'd like to talk about iGEM briefly.
iGEM is a program that was founded at MIT in the early noughts, around 2003 was when it was first prototyped. Today it stands for International Genetically Engineered Machines. And it is essentially a student-led program.
It's an annual powwow, right? Like jamborees type of.
Yeah, it is essentially a competition to do the best genetic engineering possible, and it's got a really structured framework in how the work that can be done and how it needs to be presented, and all the various components, everything including social engagement. Mm-hmm. But it is essentially to see what students can make with these tools and technologies, standing on all the other projects and tools developed by all the previous
that went before
them. So
and these are high school and college teams that compete, right?
High school and college teams. But it is really fun, collegial, and the work coming out of these projects is staggering. It now rivals PhD work.
So describe a couple of mind-boggling, head-scratching, or whatever gerund you choose, projects that you've been aware of at iGEM over the last several years, where just some of the things that kids have output.
The ones that stand out in my mind were actually from a number of years ago. For example, there was one group that made beer. And they re-engineered the yeast to make a cancer medicine. So it was literally cancer-fighting beer. And I thought, Oh, that's awesome, because I wouldn't miss a dose.
Right. Yeah.
So
you wouldn't have compliance issues with that.
Wouldn't have compliance issues, if anything. Yeah. Can you overdose on a cancer medicine? So that one was interesting. And a number of years later, another group came along and improved the entire process and actually made caffeinated beer. They made beer with different flavors, lemon flavor. There's just yeast is becoming a tremendous platform for doing a lot of engineering work. There was a group that made biosensors using pregnancy sticks. When you think about the technology that went into making a pregnancy test, it's remarkable, but it makes a great diagnostic indicator.
Sure.
And they re-engineered these pregnancy tests to be able to look for various bacteria and viruses. And it's just like
pathogens.
And it was like awesome. So you get a very clear signal. Right at the beginning, a group made bacteria behave like a photographic film. It was just black and white at the time, but if it had a light shine on it, it changed, it went dark. And so they essentially made gigapixel film using living organisms. I just saw in the news a few weeks ago that another group had engineered that same process to make color film. The bacteria actually change multiple colors depending on the light. And there's so many examples. People have made medicines, they've made sensors. The arsenic biosensor was a big one because arsenic you can't detect in water except using an expensive chemical system, and it poisons thousands of people every year.
Yeah.
But the thing that's important is this is work done at the level of PhDs or biotech companies.
Yeah.
And it's being done by relatively inexperienced people that now have access to very powerful tools and the right mentorship and support.
Now, one of the things that fascinates me is when we see this level of capability in people who are still fairly early in their educational trajectory. We look at that, and then we look at the incredible speed with which synthesis is getting cheaper and more reliable and more capable and so forth. It tells us that all of the extraordinary things that Mr. Church has accomplished as a single individual, there will be soon hundreds and then thousands and then tens of thousands of people able to execute at something like his level, not with his intellect, not with necessarily his deep understanding of biology, but empowered by tools that will enable to operate at something akin to his level. Not all of those people are going to be great folks. Hopefully the vast majority will be, but there's going to be some bad eggs in there. Knowing that and knowing how rapidly our capabilities are improving, what worries you?
Oh, well, and I've explored some of this using the media of science fiction as well. I believe that if you're really good at targeting cancer cells to kill them, you could target individuals. So there is the potential use of these programmable medicines, the same technology to do targeted assassinations.
Now, you wrote a story about this, right?
Yeah.
Was this in the Atlantic? Where was
this? Was in the Atlantic magazine. We killed the president.
Yeah.
And
in the story.
But that was to sensitize the security community, which worries about these things, that these technologies are coming. And I'm not going to sit here and design the next flu, the next pandemic. That
you will not.
No, but that's actually really hard to do because that's what nature's job is.
Sure.
It's just like it can infect as much as it can. But it used
to be really hard to read a genome. And now it's
but you
really
don't need
easy.
to read a genome. Like reading the genome gives you some ideas, targets, but it's not going to help me make a pandemic, but it could help me make a targeted bioweapon.
Yeah.
And so I believe that there is motivation and tools and technologies and resources to make targeted bioweapons. Whether we're doing it for a cancer cell or or a particular public figure. So that's one, but it doesn't keep me up at night because I'm not a target.
But let's just to finish the thought, what happened in that story? Tell folks what the bad guy did and how that pathogen released in the wild was able to zero in on a solitary person who, because it was science fiction, it was the president.
Yeah. So in the particular scenario, the people that were responsible for this, and we never really figure out who it is, has access to the president's unique DNA code, their sequence, which is pretty easy to get. Your dandruff sequenced.
Yeah.
So any pillowcase, database, anything, tapping a sewer line. It's pretty easy to get at someone's DNA, and the sequencing technology is cheap. A look under the hood. But then they designed a virus that would just spread
through the world at large.
But in this case, they knew where the president would be speaking at a public event, and they just aerosolized it in the room. Essentially, everyone just caught the flu, and the president got a bad neurological disorder.
So unlike personalized medicine, this was a personalized pathogen in a sense. So that's the kind of thing that could happen. But
but there's other ways to leverage this. Like today, we're seeing ransomware spread around the world today, where your computer just locks up unless you pay a fine. So I can think of all sorts of scenarios like that all day long, because we see it play out in computing all the time.
We do.
If any time there's a vulnerability, someone exploits it.
Yeah.
It turns out that, well, that's how it works in nature too. If there's a vulnerability, nature exploits it. So I know that we actually have the best defenses we can imagine. Our bio defenses for you and me are so much more sophisticated than our computer defenses that I don't worry about biology attacking me that much. I can imagine
even engineered biology.
Well, again, engineered biology is a double-edged sword, like all technologies. To have the technologies that would allow me to make a really powerful bioweapon that can hurt large numbers of people assumes that large numbers of people don't have access to use that technology as defense.
Yeah.
But the positive applications of this technology far outnumber the nefarious applications. And so you're actually going to see an incredibly powerful, empowered generation of people doing good.
Oh, yeah.
Before you see a lot of bad actors in the space.
Yeah.
And right now, the bad actors are produced by nature. We see this whether it's Zika or Ebola or the influenza, which kills tens of thousands of people in the United States every year. These technologies can allow us to detect those viruses faster and essentially make antiviruses and other therapies so much faster than we have today. So the positive applications to help tens of thousands, even millions of people exist immediately.
Yeah.
And should not be stopped because of some fear that a lone rogue individual is going to do something nefarious.
Oh, and couldn't be stopped anyway. You'd have to stop the entire scientific project, which simply is not going to happen.
Can you imagine Homeland Security stopping biology from getting on airplanes?
No, it just means that, and if certain vectors of research were stopped, it would just mean that they're going to happen in China.
It'll go somewhere else.
It will go somewhere else.
So to get back to what I worry about, I worry about the U.S. not being able to lead this technology. And it's not because I'm U.S. centric. I'm not. I'm Canadian.
You're Canadian. Yeah.
What I actually worry about is the U.S. has motivations I understand for the most part. They want to do reasonably good things to make money, which is, I don't mind. I understand where people are coming from. And it also has really good systems to punish people that do bad things. That isn't universal.
No.
Those fundamentals are not universal. You get people doing, if people are using this technology not to make money or do something reasonably good for people to make money, they might be doing it because of some belief system that has nothing to do with money that I just can't relate to.
Right.
You can take it, just an environmentalist that says humans are destroying the planet might want to just say, let's get rid of the humans. Now, they're not going to be successful, but that's something that's, is not out of bounds.
And, the computing analogy is actually in some ways a reassuring one, because we all know that the world has ample hackers who are very interested in taking down computers for all kinds of reasons, not just ransomware, but also for fun. Yet on any given day, the vast majority of computers do not crash. And so if a similarly nefarious entity or group turned its sights on humanity, it's likely that the good guys who would get the equivalent of antivirus software would probably allow the vast majority of people to boot up on any given day. And there was something you said in an earlier conversation, which I also take heart in, which is unlike computer viruses, which do spread at the speed of light and have not managed to take down the entire information system despite that, biology does spread at the speed of diffusion, which is much, much slower and creates much more opportunity to figure out what's going on.
Yeah, I think that our fears here are, we need to listen and understand. We should also play some games with these types of simulations, so that we can kind of understand the thinking and potential weaknesses and build defenses that are appropriate for the times. Today, I like pointing this out. When we go into virtually any facility, there are smoke detectors and fire sensors, CO2 sensors in many cases, or carbon monoxide.
Carbon monoxide, yeah.
And so there's no reason why we couldn't have biosensors in a decade in every room. And so if someone walks into an airport and they have a virus, they get picked up. And instead of being thrown into a cell, they say, look, we should send you for some additional screening. So I see the potential for just much better diagnostics and sensing moving forward. And for airborne type pathogens, things like viruses, et cetera, I think we should have universal detection technologies. Right now, we use the organisms and people getting sick as essentially the sensors. They show up at the hospital and the doctors scratch their head and don't have the right tools for figuring out what happens.
The sensor is the emergency room, and somebody has to walk themselves into it in order to be sensed.
And right now the tools are not very good for detecting most microbial or viral pathogens in the best of cases. So I think we should be using this decade to really enhance our sensor technology, because when you're blind to something, you're afraid of it. When you're in the dark, and now's the time, to really turn the lights on.
And when you said we should play with these scenarios now, I think that's actually, frankly, one of the roles of storytelling.
Yeah.
It's what you did in your short story. To a certain extent, it's what I did in this novel, which is to speculate about things that we sure don't want to have happen, to hopefully raise some alarms and some hackles and get people aware of the fact that this could be possible. And, maybe taking countermeasures long before it becomes an actual threat.
But they're really to get, to prime the imagination and think about, if that's a possibility, how do we stop it? How do we stop the person who looks, for whatever reasons, has some interest in this area and is using the tools to kind of dream up things that really shouldn't be done? How do you identify them early when they're using a design tool or accessing a database? One of the main checkpoints that we have today is between the design tools and the DNA synthesizer.
Right.
Because I can think all sorts of crazy things. It's when I hit the print button, that it starts to translate to reality. So that's a major checkpoint.
And it's a checkpoint now because synthesis is centralized. You have to get synthesis done at a central bureau or at a company like Twist. But when it becomes something that gets done in a box, that safeguard disappears.
Not necessarily, because if you had a little box that synthesized DNA, it could be adding little segments of DNA that are essentially barcodes.
And maybe flag something like, oh my God, this guy's making Ebola.
That would just be a connected online device. Just saying, this person is trying to print something dangerous, a toxin or a virus, and just not allow that just because it's a web connected device.
Well, I am very glad that you are not kept up at night about this stuff, because you are smarter about it than the vast majority of people.
I am kept up about the U.S. leadership thing. Right now, we are seeing, tremendous reductions in science and technology spending, education, in biological sciences is across the board terrible. I think this field right now is just doesn't have enough practitioners. This shouldn't be for an elite small community. This should be something that that really anyone that with an interest can access if they want to. As computing has become.
Well, thank you very kindly for your time. You've been very generous with it.
Oh, thank you for coming out to my remote home in the woods, where I try and surround myself with a bit of life.
Well, luckily, it is a delightful and very pretty drive, so I was very happy to do it.
It's always a pleasure.
Thank you. This is a quick message from Rob. Before Tom and I start our discussion of the interview, I'd like to give a brief preview of some upcoming episodes. Next week features a fascinating conversation with venture capitalist Steve Jurvetson, who's famous for backing Elon Musk in multiple ventures, including Tesla and SpaceX. Steve and I will talk a bit about Elon, but we'll mainly focus on quantum computing, a major topic in my novel that he knows quite well. The following week, we'll discuss the grim subject of terrorism with Sam Harris. Sam is one of the most prominent and controversial commentators on this topic today. He's known for his many New York Times bestsellers, his wildly popular podcast, and for a very sharp-elbowed debate about Islamophobia with none other than Ben Affleck on Bill Maher's show. The next episode will feature a chilling discussion of the dangers humanity may face someday from superintelligence. I'll be joined by James Barat, who wrote the fantastic book, Our Final Invention. And the week after that, I'll be talking to British astronomer Stephen Webb about many subjects, including why we have not yet detected any signs of alien intelligence in the universe. This is a much deeper and more serious question than it may seem on the surface, and it's one that has dozens of plausible answers, some of which could have alarming ramifications for humanity. Like, seriously. Now, because our audience is growing very quickly, I'd also like to mention some prior episodes that newcomers here will have missed. None of them have gone stale, since this podcast is not about today's headlines, but rather about ideas with staying power. So, last week, I had a terrific conversation with Cindy Cohn. She runs the Electronic Frontier Foundation, which is by far the world's largest organization defending civil rights online. Cindy and I discuss privacy and government intrusion in the digital age, a big theme in my novel and also in our world. The week before that, I discussed topics including consciousness with neuroscientist Adam Gazzaley. In his huge lab at UCSF, Adam is developing video games which could help prevent or even cure horrors like Alzheimer's and autism. One of his games is now in the final phase of the FDA approval process for ADHD. So as early as next year, we could literally have doctor-prescribed video games. I could not make that up, and I am a science fiction writer. And finally, our debut episode from a few weeks back dives deep into the science and business of augmented reality, or AR. My interviewee is the founder and CEO of Meta, which is arguably the world's leading AR company. Now, the folks at Microsoft are among those who would argue with that characterization, so give the episode a listen and you can be the judge. You can find quick links to all those episodes at after-on.com/podcast. And/or you can easily subscribe to the After On podcast on your smartphone. Instructions for doing that can also be found at after-on.com/podcast. And now, back to our episode.
Rob and I will now discuss the interview and sometimes make some specific references to the novel After On, particularly pages 180 through 241, which are officially on today's roster. So if you're not reading the book, you might get a little confused. But since you've got this far, you'll probably still enjoy the discussion. Just beware, if you're planning to read the novel, spoilers ahead. Now, unlike most of our podcasts, which touch on the issues that radiate throughout the story, this interview with Andy is pretty laser-focused on the last 3 pages that we read this week. Although it's not a spoiler to say synthetic biology will be important throughout the rest of the book.
Exactly, which made this a perfect place to share Andy's insights because the stuff is complex and really important to the storyline, and also, I'd argue, to humanity as a whole over the coming decades, and that is not an exaggeration.
Which is scary because this section ends on such an ominous note. And since she has such a lovely voice, why don't we have your audiobooks reader, January Lavoie, remind us of exactly how ominous that note is.
Rocketing out the door, Mitchell decides to find Buford's closing argument comforting. Emerging synthetic biology tools are powerful and scary. But even the most destructive crazies define themselves in opposition to something, or someone. No evil group, not the Nazis, not the Spanish Inquisition, not ISIS, has ever wanted to kill everyone. Right?
It feels like it'll be easier to keep a lid on synthetic biology's scary side as long as it remains synthesis as a service, as you and Andy put it in your conversation.
I'd say that's true, and DNA synthesis is sold as a service today, and it's how Flutter sells DNA in the novel. And this is a good safety feature because you can identify exactly what someone's making, provided it's a known organism with a remarkably short sequence of DNA.
Now, why is that?
Well, as Andy pointed out, the standard building block of synthetic DNA is currently a 100 base pair oligo, and that can appear in an awful lot of configurations, 4 to the 100th power to be exact, which is a number with 22 zeros in it. That is way, way, way more species of anything than will ever inhabit this planet. So if you're trying to create a nasty derivative of Ebola, say, each and every one of the 200-ish oligos that you would need to build an Ebola genome, which is about 20,000 base pairs, would set off alarm bells at Twist Bioscience or wherever else you're getting them printed. And in a prior conversation, not recorded unfortunately, Andy told me that DNA printing services do have watch lists of nasty things that should not be printed.
Okay, that makes me feel a little better, but it does sound like the era of centralized DNA printing services will be brief.
It will. It's hard to imagine that anyone seriously engaged in bioengineering will lack an on-site DNA synthesizer 15 years from now. It may even be rare to find a high school that doesn't have one 15 years from now.
And it'll be harder to keep an eye on 1 million DNA printers than a couple of large central companies like Twist.
You got it. Now, Andy did make the point that synthesizers will tend to be internet-connected devices, so probably they'll be rigged to report every print run to the FDA or something. But a good hacker could probably figure out a way around that.
Yeah, we thought the same thing about laser printers and counterfeiting, and it didn't exactly pan out that way. But with laser printers, you could crash an economy. With DNA printers, you can crash humanity. And what Andy describes means the number of people who could do something nasty is likely to increase quite a bit.
Yeah, and I'd say it already is increasing. In my conversation with Andy, I used George Church as a metaphor as much as anything else, but he is one of the most brilliant bioengineers alive. So let's imagine there are certain things, thought experiment, that George alone can do right now. Well, every year, some of his grad students fan out and join a bunch of different labs, statement of fact. And every year, the tools that all of these labs are using improve a lot because we're advancing down this curve much, much faster than Moore's Law. And we all know how transformative that was. So in a year or two, these former grad students of George's will be able to do anything he can do today. And some of them will be training a bunch of new students. And then those kids will fan out. Go out a few years, the tools will literally be 1,000 times better than they are now. And all the ingenious things that George alone can do today with chemical synthesis, CRISPR, and nanopore sequencers will be embedded in a $25,000 box that 1,800 labs have. This is all a long-winded way of saying that the passage of time makes wizards of us all. They're stocking stuffers out there that Thomas Edison couldn't even have dreamed of. And the kids operating them on Christmas morning are generally less smart and capable than Thomas Edison was.
Now, when I was in 8th grade, I could mix a couple of chemicals at home and create a nice little strand of nylon. Someday soon, 8th graders will be able to press, print, and destroy the world.
Well, the good news is, no, that probably won't happen. But the bad news is, I say it probably won't happen because somebody else will destroy the world long before it gets to 8th graders. It could be the next Jim Jones. It could be a nihilistic group that likes blowing people up, or a next generation Columbine kid. Lots of people will get their hands on this stuff before 8th graders do, and some of them are pretty nuts. Now, none of this is inevitable, but it's not a danger that will just magically resolve itself before we invent the capabilities that make it a danger. So we need to think about these things now. We need to freak out about them now, because hopefully freaking out about them now will motivate us to work on solutions way in advance. And there's precedent for this. I mean, the world freaked out about totalitarianism after reading 1984. And at that point in the late 1940s, it would have seemed that totalitarianism was pretty much inevitable on the left or the right because of recent history. The world freaked out about nuclear winter after reading and watching all kinds of stories. And while neither of those dangers is entirely in the past, I'd say we're doing a lot better relation to either of them than most smart people expected back in the 1940s.
Now, I am sufficiently freaked out.
Good.
But before I go live in a bubble in Idaho, up in the mountains, I want to point out that Andy didn't seem anywhere near as worried about this as you.
He isn't. And as I said in the interview, this gives me a huge amount of comfort because he knows so much more about this stuff than I do. But even if he's almost definitely right, and I sure hope he is, the consequences of being wrong are so severe that we have to take them seriously. It's like the umpteenth safety check that pilots make before takeoff. Planes are so well built, they're almost definitely wasting their time. But the downside of skipping the pre-takeoff checklist when you shouldn't as a pilot is devastating enough that you do it every time. Now, one advantage that we've got is that biology is defense all the way down. Every organism on this planet descends from millions of generations of critters that someone else wanted to eat, infect, or otherwise kill. So to use one example, that's not the case with computers. As Andy and I talked about, they descend from generations of expensive, precious things that people took very careful care of. So they're pretty vulnerable to malicious hackers and viruses. And despite that, on any given day, the vast majority of computers don't get infected and don't crash. So maybe we, with our bodies that are built to survive unceasing attacks from birth, from the tiniest cell on up, will be tough customers when evil biohackers start trying to make us crash.
I like the idea that what we don't know about how our bodies work might be the thing that eventually saves us.
It's true.
But let's move on from the scary stuff. At the very, very beginning of After On, some of the promise of next generation biology was mentioned. And since we all really like January Lavoie's voice, let's have her read about it.
So yes, Silicon Valley did give us Farmville. But across the decades, its countless startups have also rebuilt our world's foundations. Some relentlessly advanced the microprocessor, enabling the digital wonders of our era. Others honed DNA sequencing, which cracked the human genome and will one day help to cure cancer.
Well, that sounds a lot better to me than an 8th grader killing us with a print button. So, how could DNA sequencing help cure cancer?
Well, there's lots of ways, and I'm going to focus on one that I know very well because it's being developed by a company that a good friend of mine started. The company's called Grail, as in Holy Grail, and it's kind of a spin-out of Illumina, that dominant maker of DNA sequencing machines, which Andy and I talked about. Now, Grail got started because of an awful tragedy, which is that my friend's wife died young of cancer. And like so many cancers, hers was asymptomatic until it reached stage 4. And when a cancer is discovered that late, there's an 80 to 90% chance that the patient won't make it. Whereas, even with the blunt instruments that we have for curing cancer today, if it's caught at stage 1 or stage 2, the odds invert. And there's an 80 to 90% chance that the patient survives. So when Laura passed away, my friend Jeff was running the life sciences group at Google X. And he'd also been on the board of directors of Illumina for quite some time. So after, taking significant time out for grieving, he looked at the Carlson curve, which is the curve that shows how fast DNA sequencing is getting cheaper. And he looked at it with knowledge of what was in Illumina's pipeline and its next generation and next generation technology, which as a board member, he had a pretty good understanding of. And he realized that within a few years, maybe by 2023 or so, maybe 2024, maybe 2022, but soon, it'll become possible to do a simple blood draw, like when you're getting a standard blood test, and detect almost any form of cancer that might be hiding in your body unbeknownst to you at stage 1 or stage 2.
Wow.
And this test could be cheap enough. And in fact, if you trace out the Carlson curve and you understand Illumina's pricing, will be cheap enough to be part of anyone's annual physical fitness exam. And it will, assuming that works, and obviously the work is just beginning, save millions and millions of lives per year by catching cancers at an early stage when they're largely curable rather than later when they largely aren't.
That sounds fantastic. It fits in with what you guys were talking about of the ever cheaper ways of doing this. But how would gene sequencing do that?
Well, even with very early stage cancer, apparently tiny traces of the tumor's DNA will end up in your bloodstream. So if you have a DNA sequencer that's so powerful, it can actually read a huge percentage of all the nucleic acid in a simple blood draw. You will detect that tumor very, very early. In fact, I have at times heard Jeff using terms like stage -1 and stage -2. That won't be, I'm sure, the first version of the product, but really early, getting earlier and earlier and earlier as the technology continues to get better, as it will. Detecting the tumor when it's tiny and long before it's had any chance to metastasize. And there also will generally be markers on the DNA that will indicate where the tumor is located, which is important once you know that it's there.
All right. Now, can this really just be 5 or 6 years off?
That's how it looks. Obviously, there's a lot of development that needs to happen, but the company has already raised over $1 billion from very smart people like Jeff Bezos and Bill Gates, and also from several of the largest pharmaceutical companies in the world.
Well, I would like to make sure that happens before me or anyone I know and love has to deal with it. So I'm hopeful. I'm very hopeful for that. And on that happy note, I say we call it a day.
I agree.
But do join us next week when we'll be talking to VC Steve Jurvetson about quantum computing, which will be at the heart of the second half of the novel. And at the end of that podcast, we'll be discussing pages 241 to 285 of After On.